Okay, anyone missing anyone? Let me try. That is hoping. Haha. I'm so proud of my electronics engineering knowledge here. The interference stopped when I put the power cord away from the HDMI cable. You'd hope they're insulated well enough, but I guess the frequencies are so high. That aside, seems to work. Maybe sound still works for the videos to come. Let's start. Welcome back to Algorithms of Bioinformatics. We'll start a fresh new topic with some, I think again, very nice puzzles and algorithms to solve them. This time we're putting together genomes, something that popped up, or I've alluded to this human reference genome and so on, but we didn't really talk about where these come from. And today I want to show you both a little bit kind of the intuitive version of the lab techniques so that we can have a bit of a feeling for what kind of restrictions they come with. And then again, as always, focusing on the algorithms to work with what comes out. And let me start with a little fun motivation for this problem. And this is from Compo and Pefzner's book. Suppose you take a stack of newspapers. These are all identical New York Times issues from whatever, June 27th of 2000. I'm not sure what happened on that day. Maybe someone can look that up. They don't give away the solution there. Now, suppose you take that pile of newspapers and you just blow it up. What you're left with, and suppose that paper doesn't burn. So maybe their example isn't perfect. Paper doesn't burn. What you're left with is all these little snippets of these many copies of the same paper. And from that, you're trying to answer what was the headline on that day, which I indeed now am curious to find out. Clearly, the example is a bit whimsical and extreme. Now, how would you even start having any chance of doing this, right? If you take a single newspaper and you rip it to pieces, or maybe a more true version, you put it in one of these machines that just carefully cut it into very tiny pieces. If you just do that, and all the pieces are nice rectangular, as these machines do, there's no way to really assemble back the original paper, because maybe there's a few cases where some character sticks over the edge and you can make sure that these are connected. But oftentimes, or between the lines, there will be nothing to guide you. So there's just not enough information for reconstructing this. The key why here there is a hope is that you have these many copies of the paper. So likely they're going to get scattered in different places. And so by finding pieces that have the same information from different copies and overlap them, there's a chance that you can puzzle your way through the entire paper, maybe. And unfortunately, this is not so far away from what genome assembly looks like. You'll see in a second, there's much less dynamite and boom involved. Apart from that, it looks almost as daunting. So let's start with a bit of the lab technique background. How do we even start reading out something that is, 3 billion letters long, but so tiny that you can't see it, you can't touch it. Even on a microscope, you can see chromosomes, but not much more fine-grained versions. How do we do this even on a conceptual level? And the answer is through clever indirect tricks and chemical tools at the end of it. There's two main methods, and I want to briefly present both. The first one is the historically more relevant one that was, my understanding, is the main one used for the human reference genome. And then since the turn of the millennium, roughly, this was replaced by another technique. But this first one is kind of old, and the principle hasn't changed so much. The only reason why it fell out of fashion is cost. I think it was when they started around pick your favorite currency in unit per base. So sequencing one base costs one euro, and now multiply that with the 3 billion bases, and you get a hefty price tag. It's also relevant that he won the Nobel Prize, Frederick Sanger, that is. And it was his second. There's not so many people that got 2 Nobel Prizes. He's one of them. He popped up early in the lecture for sequencing insulin. And the other one, again, a sequencing task, but this time nucleic acids. He was pioneering this a lot.
Second type of oligo on the surface.
Nope, that's not the one.
DNA sequencing is the process of working out the order of the building blocks, or bases, in a strand of DNA. Before we can sequence the DNA, it has to be cut up into smaller pieces that are inserted into plasmid DNA and then put into bacterial cells. This makes it possible to produce lots and lots of copies of it as the bacterial cells multiply. The DNA is then isolated from the bacteria and sent for sequencing. The isolated DNA is transferred to a plate where the sequencing reaction will take place. A mixture of ingredients is added. These include free DNA bases, A, C, G, T, DNA polymerase enzyme, and DNA primers. Modified DNA bases labeled with coloured, fluorescent tags are also added. These are called terminator bases. To start the sequencing reaction, everything is heated to 96 degrees Celsius. This separates the DNA into single strands. The temperature is then lowered to 50 degrees. This enables the DNA primers to bind to the plasmid DNA. The temperature is then increased to 60 degrees, and the enzyme DNA polymerase binds to the primer DNA. DNA polymerase starts making a new strand of DNA by adding unlabeled DNA bases to the target DNA. It continues to add DNA bases until a terminator base is added. These terminator bases have been chemically altered so that no more bases can be added to the new strand of DNA. Once a terminator base is added, the DNA polymerase enzyme stops making DNA and falls away from the strand. Everything is then heated to 96 degrees Celsius again to separate the new DNA strand from the original strand.
So maybe a quick kind of summary up to this point. The first step was how to even make many copies. Remember, we have the pile of newspaper. We need many copies of DNA. And it seems to be the easiest is to hijack bacteria to do it for us. Kind of using life itself to do it, which has a potential problem. What if some small snippet of the DNA you want to copy is somehow lethal for bacteria for some stupid reason? So you may have gaps because of that. I think this is what happened at some early stages. Okay, but first ignore this part. There's some way to produce copies of DNA. And then the second one was we start reading at some specific places. So we need these primers. We can only start reading at specific points. But apart from that, we get copies of parts of the DNA of random length, because it's random when this terminator base finds its way into the process of creating the copy. So you get many copies from starting from the same place but ending in random positions. But it's important that you can control where they start. That's the primer they call it. That's a special sequence where this reverse complement copy can attach to. Okay.
This process of heating and cooling is repeated again and again to produce lots of fragments of DNA of different lengths. The length of each fragment depends on when a terminator base got added. To read the sequence of the DNA, the various fragments are separated by length using a process called electrophoresis. A capillary tube is lowered into each well of the plate and an electrical charge is applied. This causes the negatively charged DNA molecules to move through the capillary tube. Each capillary contains a porous gel. The shorter fragments of DNA move through the gel more easily than the longer DNA fragments. As a result, the fragments become arranged by size from the shortest to the longest. As the DNA fragments come to the end of the capillary, a laser makes the terminator bases light up. The color is detected by a camera and recorded. Each terminator base is labeled with a different color. A fluoresces green, C blue, G yellow, and T red. The shortest DNA fragments will be read first and the longest read last. The sequencing machine records the color of the terminator bases as a series of colored blocks. Each colored block represents the labeled terminator base at the end of each fragment of DNA. By converting the colors into letters, we get the sequence of our piece of DNA.
All right. So a pretty clever trick, this sorting by length. Question?
Just so you know, on the 27th of June 2000,
Ah.
front-page news at the New York Times was an article announcing that
The Human Genome Project.
scientists had completed
course.
a rough draft of the Human Genome Project.
I should have made the connection. The year 2000, I remember. Yeah. Yeah, there was some controversy at the time that people, the science news cycle spat out, the human genome is deciphered, when it was only really 90%. And I think it took, it's only a couple of years ago that they finished it in entirety because of these problems that pop up here. So I'm not 100% sure, but I would assume that this last step is really a bottleneck, right? You have to get these molecules moving through the tube. You can't do that arbitrarily quickly because you need to have the light recorded carefully. And so you need, essentially, for each sequence, you need a tube. And I could imagine you can build machines that do this somewhat in parallel, but maybe not millions of things in parallel. That's why there's probably an inherent limit to this. The benefit seems to be that you can sequence relatively long reads. Maybe 1,000 bases is still okay. The downside, it seems to be cost, and potentially also sometimes you get errors that the readout is not perfect. Well, perfect will never work. But the second group of methods seems to do really well on reading much, much shorter reads. Think of 150 to 200 base pairs or bases. But at a tiny fraction of the cost and very reliable for this first part at least. And then once you get to longer reads, it becomes very quickly noisy. Now this is commercially available since 2005. So they were developed after the Human Genome Project was over, and I think that's not a coincidence. Because with this reference genome, there was suddenly a market for sequencing genomes and doing something productive on it, all sorts of health applications where people could again make money with the application. And so I want to show this video, take into account that this is by Illumina sequencing themselves. It's one of the leading companies that builds these machines. So there's a little bit of advertising in. They might not stress the weaknesses as much as maybe some other videos would. But I think it's very nicely illustrated. For this one, I think the details are a bit more messy, and we don't care about the details all that much. Maybe just take it as a sign of sophistication, that people really developed iterations on this just to make it more parallelizable and more reliable.
Base call. Illumina sequencing systems have moved from a 4-channel detection.
Let's start this from the beginning.
The Illumina sequencing workflow is composed of 4 basic steps: library preparation, cluster generation, sequencing, and data analysis. There are many ways to prepare libraries based on sample input type and sequencing application. All library preparation methods add adapters to the ends of the DNA fragments. Through library preparation, these adapters contain regions such as sequencing initiation sites, indexes for molecular barcoding, and regions that will bind or hybridize to complementary sequences on the flow cell. Clustering is a process where each library fragment molecule is amplified to create a cluster of identical copies on the flow cell surface. Illumina utilizes 2 flow cell architectures: random flow cells with a lawn of oligos over the entire surface, and patterned flow cells with oligos only in nanowells. Newer Illumina systems utilize patterned flow cell technology to create clusters at greater density and efficiency. Flow cells are coated with a lawn of 2 types of oligos. Cluster generation occurs when library fragments hybridize to the surface of the flow cell. DNA fragments are bound to the flow cell substrate through hybridization of complementary sequences. After libraries are attached to the flow cell, the strands are now clonally amplified through a novel amplification chemistry called exclusion amplification, or XAMP. After the polymerase creates a copy of the hybridized template molecule, another copy is made by hybridizing to the second type of oligo on the surface, creating 2 identical copies tethered to the surface of the flow cell. The process is then repeated.
So my understanding is that this is how they trigger a chain reaction so that you get the same snippet of DNA in each of these clusters. I think they don't really clearly say this, but there's a single one attaching to these these fragments at the bottom. They stick to any kind of DNA that has these adapters attached. And they hope that one will stick and then will amplify so that the cluster is filled with copies of the same read, or the same sample.
Repeated many times per cluster, resulting in clonal amplification of all the library fragments. XAMP performs this process quickly, efficiently, and in real time. After amplification, the reverse strands are removed, leaving only the forward strands. The 3' ends are blocked during the removal of the reverse strand to prevent unwanted priming during sequencing. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, a mix of fluorescently tagged nucleotides are available for addition to the growing chain. Only a single fluorescent base is incorporated that is complementary to the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called Illumina sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. Illumina sequencing systems have moved from a 4-channel detection chemistry to a 2-channel detection chemistry. 2-channel detection chemistry improves efficiency and reduces sequencing runtimes. New XLEAP SBS chemistry further improves the speed and accuracy of the 2-channel detection approach through an improved block and linker and a new polymerase engineered to incorporate nucleotides faster and with higher fidelity. This delivers up to 2 times faster incorporation rates relative to standard SBS. All identical sequence strands in a given cluster are read simultaneously. Hundreds of millions, up to billions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step of the example, the Index 1 read primer is introduced and hybridized to the template. The index read is generated similar to the first read. After completion of the index read, the read product is washed off. The template now folds over and binds the second oligo on the flow cell. Polymerases extend from the second, reverse strand oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized, and the original forward strand is removed, leaving only the reverse strand. Index 2 is read in the same manner as Index 1, with the introduction of an Index 2 sequencing primer.
Why they do this double
Index 2
reading, I
product
think, is
is
not
then washed
exactly
away.
clear. They don't explain it.
Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. This entire process generates billions of reads representing all of the fragments. Sequences from pooled libraries are separated based on the unique indexes introduced during the library preparation. Based on the library preparation and chosen sequencing application, downstream processes can either assemble sequences to reconstruct the original sample without the need of a reference sequence, map and align sequences to a reference genome to identify regions that differ from the reference genome and detect variations, or count molecules via sequencing reads to assess abundance and gene expression. This omic data can be easily centralized within the Illumina connected software ecosystem.
Okay, we'll skip the advertising part about their software infrastructure. You see, there's a lot of steps that they don't actually explain what happens or why. Everything is read forward and backward. Maybe you get better errors, fewer errors that way, because you can probably sequence more carefully at the beginning of the process. I think the key point is, unlike in this previous section, you skip this step of sorting the copies, or you're producing copies of different length, but rather they have found a clever chemical way to have these bases attach with this fluorescent light, so they can read off the current base that was added, but then modify the base so that then the copying can continue. And again, the video doesn't tell us how they do it, and they may not let that knowledge out. That's their secret. But the main point is that they can do this on this whole plate for millions of sequences in parallel. The whole process is probably still slow. Each of these rounds of adding one base, reading it out, washing it off, and all this probably is seconds and minutes, maybe hours. But because they can do it for an entire large samples of millions of reads, the cost per read is very, very low. Around the point that sequencing your genome is less than €1,000 these days. And that probably includes the post-processing, which we're going to look at. Okay, so much for the lab techniques in the background. Again, certainly you don't need to know the details, but keep in mind the core problem in both of these is you only get short snippets. You can multiply the sequence in advance, so you get the snippets in many places, but you can't expect very long, reliable, contiguous reads in one go. The only way to get cheaper is to make this in parallel, but have even shorter snippets. Yeah, depending on the technology, hundreds or thousands of bases, and these are somewhat reliable, but we'll come back to this at the end. Nothing is perfect, so all the methods that rely on perfect input data will have limitations. Now the post-processing, here's again from the book, The Idealized Picture. You have many copies of the same long string. You break this into pieces. This can be done chemically, as far as I understand. This is not a hard step. And then you sequence all those reads, and you can pretend that these reads are roughly the same length. Cutting them off at the end is actually harmless. So wasting a few bases there is not the big deal. then to sequence the original string, you would have to overlay all these snippets and hope that they are long enough and convey enough information that this consensus string that you built from overlaying them all has meaning or is at least close to the original string. I guess it's quite clear from the outset that this is not going to be possible for some contrived cases. Say if your original string is say 4 copies of some string, just repeated 4 times. Distinguishing that from 5 copies, if your reads are not long enough to stretch over many copies of S, is impossible. You can try to get information about if you can recognize the endpoints, maybe with how often you get fragments from each place. Maybe you can try to infer the number of copies. But there are fundamental limits to the reconstruction problem. It's not in general possible to get this string in all cases. That we'll have to live with. Okay? So oftentimes what people say formally is we find the shortest string that explains all those reads. Are there questions on the sequencing methods that would make sense to discuss? Or you're roughly happy with the...
During the overlap, like would the sequence repeat somewhere else to, like for example AGAA, maybe it comes after, like at the end instead of like that particular sequence, that green color sequence?
Yep.
Couldn't that be at the end as well?
For this particular one, it looks like no, right? Because it would have somehow the TGA. But in principle, what you say can happen. It could be that the same short substring occurs several times, and then it's not uniquely clear where a read goes. In practice, there's 2 answers to this. One answer is, yes, that happens, and it's an actual worry. If the sequence that's repeated is very long, then we don't know for sure how to solve this. There you need methods after the results from this chapter to manually fix it. In a way, if you have a long repeat, probably still what comes before and after will be substantially different. So from the starting points of the repeated region, you can maybe figure out that it occurred 2 times or 3 times. So there's ways to deal with that, but in principle is a problem. The other answer is there are many repeated sequences that are shorter. So they're short enough that you can, with some reads, cover them entirely. And then it's less of a problem because you see the changing context. That is why the length of the reads is kind of has a big influence. Many of these repeated regions, there's one whose name I forgot that occurs 1 million times in human genomes, but is only around 100 bases long. And that seems to be annoying for the computation, but it's short enough that you can deal with it by covering it with reads. Let me in the next section show you a formalization of the problem where we first ignore some of the problems, and we'll come back to them a little bit at the end. And you'll see even the resulting problem is still kind of juicy enough, or hard enough to solve. To make things easier, I'm making an assumption that the fragments of the genome I get are covering every possible starting position. All of this module will use k-mers just because the bioinformatics people tend to use this. It's just a length k substring. But I think because they got annoyed by saying length k substring 1 million times, they say, oh, it's a k-mer. But it's nothing fancy. Then if we're given a text, say our genome, and some length for these k-mers, then the input to our problem will be this. The k-mer composition of a text is just all these contiguous length k substrings that the text has. So every possible starting position, you take the substring of length k, all the k-mers in the string. Now, as indicated with these double braces, in general, that's a multiset, right? You can have repeated parts, and that would mean that some k-mers might occur more than once. So if you're given a text, it's of course easy to produce this k-mer composition, just listing all the substrings. The problem that we're trying to solve is, of course, the reverse problem. Okay, here's an example just to make sure. Here, I've sorted them alphabetically to make it a little less obvious, or to show the duplicates more clearly. There's a stretch of, thank you. Come on. There's a stretch of 4 Gs. So I'm going to have the first 3 and the second 3. That's why GGG occurs twice, and so on and so forth. Life gets interesting when you try to reverse. Someone might promise you, and that's our assumption, that we get a multiset of these k-mers that comes from some specific text. And then we're trying to reconstruct that text. This is easier than the biological motivation problem for several reasons. One, we don't have sequencing errors. But 2, also the assumption that we have a k-mer from every possible starting position and exactly once will help us a lot in kind of removing some ambiguity. I'll come back a bit later to motivate parts of why this is not completely hopeless, even in the data from applications. But for now, let's ignore this and just take all the possible k-length substrings exactly once. Okay. All right, let's look at some examples. So here's a composition, a 3 composition of some text. I'm going to give you a hint. The number of k-mers is 5. So if I want 5 things in here, there's 0 up to n minus k. And n minus k is included. So the number of k-mers I should have is always n minus k plus 1. So if k is 3, I can work out that from 5 k-mers, if this is 5, what's my n? Also 3. The other way around, 7. Okay, people are puzzled. Can you figure out what molecule this is? What's the text that produced this? It's again sorted alphabetic, so don't interpret the order in any way. TAA, T, G, TGT. There's a letter missing. Hold on. One more. Is this the right length? 7 should be. Yeah. Okay. Did we use everything exactly once? This so one's there.
GA has to be the start because there's no other one having GA in the last one.
Yeah, seems to match. Do you want to describe how you approach this? Or it's a small enough example you just try randomly puzzling?
So the first one is GAA and it has to be
overlapping AAT.
Say it again?
First one is TAA, then overlap with the AAT.
There's only one double A that has to be in correspondence. That's right.
And there's no GA in the last 2 characters.
That's right. So this has to be the start because there's nothing that matches on the beginning. And I think double T signals this is the end, and then kind of greedily just assembling the thing works. Okay, so easy problem. This is just the summary of what you also produced. And here's the solution again. Now let's do a slightly bigger one. And it would be kind of more revealing if you had a bit more time to puzzle with this. And if this was kindergarten, I would have cut out the things and we could puzzle exactly, right? But you already may know where this is going. But let's just try the same strategy, right? You can work out that maybe TAA is a good starting point because there's nothing that ends with TA. Is that right? It looks like it. So there's nothing that ends with TA. So we found our starting K-mer, 3-mer. Good. Now I have to find something that starts with AA. Okay, it's also just one option. And then something that starts with AT. Now, because they're alphabetic, there's a whole bunch of things starting with AT. Okay. So far, there was no choice, really. But for the next step, so here there's several things that start with AT, but they're duplicates. But for the next one, the TG, you have options, 3 options. And now it's not clear which one we should take. And it's also clear that not all 3 will be correct. And so if we say, for example, we pick this one, then you see you can continue for one more step, but then you're stuck with the end TT. There's nothing that starts with TT, so while this is the beginning, this has to be the end. It's a coincidence that they're next to each other. But here we're done. We can't continue, but we haven't used nearly all of the K-mers, so this can't be the original text. So somehow this choice was wrong. So we could go back and say that was the wrong choice. Let's take another turn in this maze. And then let's say we do that. So at this point, after ATG, we now choose the TGC, not the T. Maybe that's the right choice. And then you continue. And again, you arrive at the end. And now it looks better. But if you watch closely, we're still 1 nucleotide too short. There's another one where we could have, maybe you see which one's missing. It's actually one, the triple G. And it's kind of clear that it has to be something like this because I have to be able to insert this one extra thing without disrupting the whole thing, right? This is almost just possible with something self-similar. Anyways, so here you could have done the GGG first, and then you're happy. Now, of course, then that's a solution. Now, in general, you may already see it could be tricky to make the right choices. We need to somehow globally look at this. Okay, but at least the puzzle seems a little more manageable with this assumption that you have this full K-mer composition. Now, we would not like to systematically not like to keep trying dead ends. We would like a systematic algorithm. So what could we do? It's a small group. Maybe you want to make some suggestions out of the blue. What would be your gut feeling would work? And if you've seen it already, like, okay, forget it, but
Similar to the first algorithm we did, maybe we can start with the start by finding the first, like the start and the end.
That's right. Starting beginning and end is always a good idea. Now, in general, is it clear that the beginning and end is uniquely recognizable? Maybe not, right? There's no guarantee that the beginning one can't also occur somewhere in the middle, maybe as a duplicate. Like, for the sake of argument, say the text is just all A's. All the K-mers are all A's. Now, this is easy because they're also the same. One of them will be the starting point. But I guess you can tweak the example. But still, you're right. If there's a unique starting and end point that we should use, sometimes there isn't. The other piece that we used repeatedly was we have one K-mer. Now, what are possible next steps? They have to overlap apart from the first and last symbol. So that looks like an edge, right? I can connect two K-mers by an edge if they are overlapping in the right way. And then I have edges, so maybe something of a graph is a good idea to represent the data. And let's maybe just try that before we think about the problem. I just wanted to throw that one in. The kind of puzzle we were doing, the tricky bit here comes from having choice, where several things match in the same way. And that comes essentially from having this substring repeated in the original text. And the same is true if you do ordinary puzzles, but puzzles where you don't have the shape of the pieces telling you much. Then this is kind of easy if the pieces are all uniquely, if there's only two two pieces that match across an edge. If you have patterns like in this puzzle, where there's many patterns that occur many times, then it seems much harder. So hard, indeed, that apart from the creator of the puzzle, nobody has ever found the solution to this yet. And there was a substantial prize money attached to this. And essentially, people gave up at some point. I don't know if the competition is still open. The puzzle still exists. And the inventor claims there is a solution. Let's suppose we believe that. So these puzzles with repetitions can get very, very hard. Okay, so maybe we should manage our expectations here. Let's try this idea of edges and graphs and see where that leads us. I'll call it the overlap graph for the obvious reason. Let's formally define again what I said already before informally. You're given such a k-mer composition. Now this you can actually define whether or not there is a single string that this comes from. Let's just say some multiset of k-mers. Then we define a graph where the vertices are essentially these reads or these k-mers. I gave them lowercase letters here so that we have a separate vertex for all the k-mers, even if they're duplicates. Okay? So I'm always going to have as many vertices in this graph as there are entries in the input, but there might be several that have the same k-mer associated with them. Is that clear, the distinction? So each of these vertices, we will label them with the k-mer, and so there will be several vertices potentially with the same label, but I want them as vertices to be distinct. And then we have an edge. This is a directed graph, because again, like if you have some k-mer like this, the orientation of the edge should go from this guy to that guy. It's a directed relation because there's an ordering. So there's a directed edge from Ri to Rj, exactly in this case. The last k-1 letters of Ri match the first k-1 letters of Rj. It's just formalized what I just said. And here's an example. Again, this is taken from the book because they drew it so nicely with colors that I felt it's not much fun in me redoing the drawing. So here's an example text where this comes from. The first step, this is just the backbone. This is the edges that are obviously there from what we have in the text. Now, the graph we defined, of course, will have more edges because there's some examples. For example, this one starts with AT. This one ends with AT. So we should have an edge like this as well, right? And there's a bunch of others. Okay, this edge is drawn down here. But there's a bunch of other edges that come from this same overlap. Now, if I have the graph given like this, it's obvious how I can read off the original text, but that's not really what it looks like. You should rather think of the ordering of the vertices being uninformative, say alphabetic, and then is a bit more messy. But as a graph theoretic notion, what are we looking for here? What is corresponding to this chain of letters where if I read them from left to right, for the first one I write the entire 3 letters, but for the next ones I always just read the last letter and add it. If I do this here along this chain, then I get the original text back. In graph theoretic terms, what would I need from this graph? Yeah, so first of all, a path, right? I have to follow edges to construct something that nicely overlaps. And I want a long path, essentially a maximally long path. I want a path that contains every single vertex, right? Because I want every single k-mer to be used in the construction of the text. So these things are called Hamiltonian paths. So in this overlap graph. In one way, that's good news. If something has a name, you can Google the algorithms for it. It's also bad news because we already know that this is a hard problem. So we kind of reduced our problem, if you want, to finding Hamiltonian paths in this overlap graph. This should be GO. And here's an example in the non-linear arrangement. And I think a nice if you follow through the edges, you'll see that this is a different text that also matches the same k-mer composition. So even in our perfect world, where we assume that our input is a clean k-mer composition of a single text, it can happen that the graph allows several Hamiltonian paths, and the problem is not uniquely solvable. We'll have to live with that. There's essentially some limitation of the kind of data we have. Again, this is not so much a concern for practical inputs. The reads tend to be so long that this rarely happens. Okay, so this is little problem. Result may not be unambiguous. Bigger problem, we identified that what we want to solve is an NP-complete problem. So there's not going to be a black box solution. Okay, maybe this wasn't too surprising. Why not? GGG says the definition of the overlap graph. I mean, no Hamiltonian cycle can ever use this loop, so you might as well leave it out. Fair enough. The definition of the graph says I have an edge. So, is a fair point. Like, this self loop will never ever be used. Not in this case where we have a single like duplicate k-mers each get a vertex. So, yeah, it's never going to be used. So again, we've ran into a hard problem, and that even ignores how to compute this graph. I guess it's clear that this is not an NP-hard problem. You compute for every pair of k-mers, whether they overlap or not. So it's quadratic in the number of k-mers. That's not, from complexity theory perspective, a bad thing. In terms of the application, it is a bad thing because you have lots of these reads. But okay, we also have to solve the Hamiltonian path problem. So we're in double trouble. Shall we do a little break? Or you want to see what comes next? Let's do a tiny one. And if you have spare mental capacity, think about what you would do. I'm going to give you the hint that there's another way to interpret the problem that leads to a different task, and that's going to be easier to solve. But in all fairness, people for decades went via this route, kind of dealing with the NP-hard problem. And maybe there's more technical biological reasons why the other route is indeed problematic. But yeah, this is early 2000s, the next observation. You're shocked. Maybe. I think the knowledge about De Bruijn graphs may have been very niche.
I received this as an Olympiad question, and it was kind of, at some point, basically.
But that's now, right? Who knows?
I mean,
I don't know the backstory about
I'm sorry to say that
this discovery.
you could have done it with things that people knew with, I don't know, 17. I don't know when you know what I mean. Like,
Sure, with the right definition, it's no magic, no wizardry involved.
I don't know. I mean, field faults or whatever. Not that.
Well, you know, making the right connections sometimes.
Yeah, no, I agree. I am just shocked.
Yeah.
This seems to me like something that's maybe 1940s, 1950s or so. Of course, they didn't.
Yeah, I
know
think
the-
this is De Bruyne's original paper is 1940s.
Right, like that's when it seems to me to be the right time, right?
Yeah, people started to get interested in these discrete problems more. This kind of
Chalk's nice.
Pardon?
Are we having break now?
Yeah. For another 3 minutes. Yeah.
Chalk's very nice.
I agree. Yeah, I've used it a bit in the chemistry lecture rooms. Sorry, you have a question?
Yeah. So, sir, actually, I want to ask if this course can be taken as application area module.
Indeed, it can only be taken as application as practical module. Sorry,
Yeah,
not
practical
application.
Okay.
It was intended to be a theory module, but there was a mistake in updating the module handbook.
I just saw the course module guide, and where I saw because
It's
I had
listed as
registered
practical.
an NLP,
Yeah.
and now I have to complete the practical or application modules,
Yeah.
at least 18 credits as part of my module guide. So I thought I should ask you. So is there any chance that I saw that 5 exercise sheets have passed already,
Yeah.
but like 7 are remaining?
Mhm.
Is there a still chance that I clear all the remaining and still be able to manage to admit in the exam or not?
I mean, out of fairness to the other groups, I would only allow this if there's some good reason why you couldn't start early.
Yeah, because the problem was that I thought it is advanced module.
I think a reasonable reason is you didn't know about the module.
And
That's a somewhat fair
but
point.
It was like advanced module. So, but in my module guide, as I understood, was that I could only take advanced modules until 18 points, which I had already cleared. The advanced modules.
Right.
But the question I asked is that it is the application module, and you said that yes. So this is why I can take this course now.
So I'm always struggling a bit with the correct terms, and I don't know which the correct terms in the English translation are. Whatever in the module handbook is listed is what we will
so it
live
is
up
both
to.
written as advanced
Yeah.
module and practical module as well.
If you can have this as a module in your study plan, that's a question for either examination office or Christoph Bockisch.
Yeah.
But it counts as a practical module, even though the content is not really is
Practical.
closer to a theory module.
Mhm.
Advanced is Aufbau und Specialization is Vertiefung.
Right.
The-
So advanced sounds a bit more advanced, but it's actually the bachelor's modules, right?
Exactly.
It's maybe
So
Yeah.
there was a limitation actually that we can only
Unfortunate
take advanced
translation.
modules of 18 CPs.
Exactly.
Sure.
So I had thought that it is advanced modules, and I can't.
I don't think it is, right? It's specialization.
Never is this never is this an advanced module.
But it is listed in advanced module list as well. In the module book, which is always has mistakes because I was.
I think you also have to check the version for the right course of studies, because they sometimes differ how modules are used. And I think that can make sense. Plus, there's the different versions, so you have to look at the right one for your. This is a bit tricky, but the question really is not something I'm going to decide. So that's something you can ask examination officer Christoph Bockisch. The other question is, if you want to take the module and do the exam, you have to do the exercise sheets. So best would be to kind of catch up as much as you can there, because the group is fairly small. I think Mohammed will have capacity to look over some past sheets maybe. So try to catch up on those a bit. I think to be fair to the other groups, we should somehow have a justification why you jump in late and don't have to do the original ones.
The reason was only the confusion. So.
Yep. So I think for now, just do the upcoming sheets, and then we'll see if
Then,
maybe we can do some extra little check
okay.
informally, like yeah.
So,
Yeah, I totally see why we have certain blocks of modules that you can only take a certain number of. I wish it was a bit easier. Like distinguishing bachelor's and master's modules, fine. Do we need the distinction between theory and practice modules? Not sure.
Well, it isn't too strict about that. Because you
In what
just have to
sense?
take
There's a
one-
minimum from both categories.
Yeah, but isn't just one of each?
The minimum is 1. Is the number of credits, I guess. Well, these things I always have to look up. Well, either way, most of the time people don't have trouble, but it popped up for this module a few times, and it's a bit suboptimal that there's this mismatch between the intended material and the official denomination.
Have we settled for a date for the exam?
We'll do oral exams.
Right.
And so there's not one fixed date. Basically, I'll see when I'm around in the examination periods, and then you can pick a date from those slots.
Are you thinking around February start?
Well, we'll have to stick to the examination periods for like a have to. It's on record, so we have to. So that's this 3-week period. So somewhere in that frame. Yeah, under special circumstances, sometimes you can also do it outside, but it's tricky because you have to register for it, and so you have to register for the right period and so on to keep life a bit easier. It's not a ton of people, so I think we'll manage to find slots, but it'll have to be somewhere in between your other exams. Okay, and turn that back on. Maybe somewhat slightly surprisingly at least, even though this overlap graph leads to an NP-hard problem, Hamiltonian path, that comes out of our sequencing a sequence assembly problem, there's another way to look at the same problem that does allow a polytime solution, and actually a very efficient one. Let me tell a little bit of the backside story, and this is, I think, 1940s is the right time frame for this. I'm going to butcher the Dutch name, Nicolaas Govert de Bruyne. The last name is pronounced De Bruyne, I learned, but I'm not sure about the first name. He pondered the following puzzle. Suppose you want a single string that contains all possible length k strings over zeros and ones as a substring. How long would that string have to be? Now, why he wondered about this is a different question. Fair enough, it's a math puzzle. And I mean, certainly there is such a string. You can just concatenate all the 2 to the k strings. So upper bound, very clearly 2 to the k times k, right? Write them all one after the other. That's hardly going to be the shortest because they're all using the same characters over and over. You can reuse characters massively. But that's an upper bound, so it's certainly possible to do this. The question is, what's the shortest, and how can you find one? Right? As any good computer scientist, if you see a new good puzzle, you try out small values. So here's the smallest sensible value, I guess. For k equals 2, there's 4 different strings that we want to see. And now let's do some lower upper bound business. Let's do some lower bound. This 2 universal string, that's how he called those. A string is universal if it contains all the possible k-mers. It needs to have at least 5 letters. Why? Every possible of the 4 needs to have a different starting position because they're all different. And so I need 4 positions where things can start. But then from the last one, I'll have 1 character stick out, right? So I need at least 5 letters to make a 2 universal string over the binary alphabet. Is that clear? Turns out you can also do this. Here's one. So you have 0, 0, you have 1, 1, you have 0, 1, you have 1, 0, all 4 occur. And we've used 5 characters. So clearly this is an optimal solution because I've just proved everything has to use at least 5. Curious. But k equals 2 is tiny, right? So maybe you have to try another one. Here's for k equals 3. Now you can do the same kind of calculation. You'll have 8 different characters. They need different starting positions, and the last one will have 2 characters stick out. So that's 10. And this one has 10 characters. And you can check that every possible 3-bit string occurs as a substring. So again, we could solve it optimally, like a smallest possible length. Now maybe you feel there should be a pattern. Right? Okay, that's what De Bruijn wondered about. And he eventually proved that you can always find, so there always is a shortest k-universal string that is as short as can possibly be. And you can find one efficiently. And this is true not just for the binary alphabet, but for any alphabet, and for strings or for k's of arbitrary size. So just doing examples wouldn't get you there, but the proof technique is very elegant. And it leads to a solution to the sequence assembly problem. So what did he do? Yeah, I said this. He constructed a different graph. And that's now called the De Bruijn graph. It starts also with our reads, so our k-merge composition. Now this time we'll use, again, vertices that correspond to strings, but there's 2 differences. These are only k-1 letters each. And I'm going to compare, kind of fold together duplicate elements. So if there's several reads, several k-mers in the input that have the same first k-1 letters, and that's especially true if there's duplicates in the input, I'm treating all of these prefixes only as a single vertex. The clever part are the edges, and I'll have an edge for every k-mer in the input, and the edge connects the vertices that correspond to the first and second k-1 characters. So here's, say we pick some example. This edge corresponding to CCA, that's one of the k-mers in the input, 3 mers in the input. It starts with CC and ends with CA, so it's an edge that goes from CC to CA. Okay? Let me maybe copy this here just to have that explicit. So all the crucial information really is in the edges this time, and the vertices are just there to connect things up. Okay? Now, I should have asked you this, but it's already on the slide. In analogy to what we've previously had, there the vertices were in correspondence with the k-mers, and I needed to find a path that uses every vertex. Now the k-mers are on the edges, so I want a path through this graph that uses every edge once, exactly once. And that's called an Euler path. I'll introduce this a little bit as a recap if you've forgotten. The main thing is Euler paths, unlike Hamiltonian paths, it sounds like a minuscule difference, visit every vertex, visit every edge. There's a massive difference in terms of complexity. You can compute these very efficiently. And so we've transformed our problem into a different graph problem. We're still solving the very same string problem, but now suddenly there's no NP-hard problem involved. Of course, as a digression, this kind of means maybe that also the special inputs we get in the Hamiltonian path problem that result from the overlap graphs may not be a hard subset of all possible Hamiltonian path inputs. Maybe that's also a way to make progress. But that would at least need studying carefully what these graphs look like. Here we don't even have to. Whatever the De Bruijn graph looks like, I can always find Eulerian paths. All right, here's an example again. let me highlight one or two things. As before, in this case, where the start and end are unique to mares, they don't occur anywhere else. You have vertices that are uniquely the start and end point of any Euler path. There's 3 occurrences of ATG, the blue ones, and so there's 3 edges for ATG. Of course, there are parallel edges, so this is a multi-graph where I might have several edges connecting the same pair of vertices. Of course, these have to be parallel edges because they're the same strings. They'll go from the same source to the same target if I take substrings. So that shouldn't be surprising. What else? We still have this infamous self loop from this triple G, but this time it's crucial that we include it. So multigraph here also means allowing self loops. And it's important because we want to take all edges, right? Now, of course, the self loops are kind of for free. Whenever you visit this vertex, you just do the excursion, and then you're done with this edge. OK. Now, we can use these De Bruijn graphs also to solve the original puzzle that De Bruijn started with. You can build this graph from the set of all possible k-mers, say all possible binary k-mers, or all over a general alphabet. Then you'll have sigma to the k-1 vertices, because all possible k-1 substrings occur at some point. And for every possible sigma to the k-mer, we have 1 edge. Now, this needs a little bit of inspection, but it's not too hard to show just by symmetry, basically, that every vertex has the same in and out degree, namely sigma. And maybe just from like a small example, suppose k is 3 and sigma is 2, so we're back to the binary alphabet. And this might be one vertex. What are the in and out edges of this vertex? The out edges are all the 3 mares that I can form that start with 0, 1. And that's these two, 0, 1, 0 and 0, 1, 1. Right? And they're going to lead to some vertex that's dictated by whatever is here. But I don't even care so much. Same for the incoming ones. They have to end with 0, 1. Then I can have a 0 or a 1 in front, and that's all. So the crucial, clever insight here is the out degree and the in degrees are equal. And that will, again, I'll recap this in a sec. That will mean that the graph is Eulerian. So there is an Eulerian cycle. And so if you follow that cycle, you visit every of the sigma to the k-mers exactly once. But you've also just used that many steps. So the total length of this is going to be exactly sigma to the k characters plus k-1. And that matches our lower bounds. If you insert sigma 2 and k equals 2, then we're back to 5. That's the example we've seen before, and 10 for k equals 3, right? So in a way, De Bruijn's puzzle is solved, and we didn't even need to do much in this case. All the observation came from symmetry. The reason why this graph is balanced, same in degree as out degree in every vertex, was just out of symmetry of the construction. In the general case, when we start with some k composition of a text, it won't be, the graph won't have all possible k-mers, k-1 mers as vertices, and it may have more interesting in and out degrees. So let's briefly revisit what is known about these Euler walks or Euler cycles. I'll use these mostly interchangeably. I should probably train myself to say Euler walk whenever I talk about these. A walk is just anything that follows edges, but it might reuse vertices as many times as we want. Now, a walk in general can use edges several times as well. Here, I really want an edge simple walk. Every edge can occur at most once. And then it's an Euler walk if every edge occurs exactly once. Okay? The picture here is the old city of Königsberg, now, what's it called? Kaliningrad, which used to have these bridges, and that's where Euler was first asked, is there a way to walk across all of the bridges exactly once and come back where you started? And, well, the story has it that's how Euler kick-started graph theory. He represented the problem as a little graph, and then he noticed, nope, in this graph there is no Euler cycle, because, of course, he didn't call it like that back then. Well, he was an interesting character apparently, but not that pompous. You can easily prove this theorem. There is an Euler walk in your graph if and only if the number of vertices with odd degree is either 0 or 2. If it's 0, then there's a cycle. If it's 2, then there's a path that uses every edge. And if neither of the two applies, then there is no such thing. And you can prove both implications. I'll leave that out for now, because this is on an undirected graph. The original problem here, the ridges were not one-way streets. You could walk them in either direction, and that would have been fine. And of course, you know Euler walks from kindergarten. Not further comment on this. How can you do this systematically? Here's an algorithm that I always show in efficient algorithms as well. There as an application of depth-first search. That's really what you can do. Maybe let me just illustrate it briefly on this. On this, what in Germany is known as House of Nikolaus. You can count the degrees, and there's 2 vertices with odd degrees, so we'll have to start and end in those 2. And the goal is to draw this drawing in one go without doing a jump with your pen. That's exactly an Euler walk. You can draw all the edges without lifting your pen. So we might start here. And basically we're doing a traversal of the graph, and whenever we get stuck, we label the edges backwards. Okay, what I mean by that, let's see that we get stuck. Let me walk here. So I'm just following edges blindly. And so at this point, I'm stuck. So now I'm going to count the total number of edges here is 1, 2, 3, 4, 5, 6, 7, 8. Is that right? So this edge that I last took, where I got stuck, I'm going to label this with an 8 and then backtrack over it, as in a depth-first search. Now at this node, I could have taken another edge, so I'm doing that. Then I'm taking these, and now I'm stuck here because I've used up all the edges. Now I go back over this one and use the next smallest free number, back over this one. Here I came from here. You have to imagine doing a depth-first search where you keep these on a stack. So I'm remembering them now because it's a tiny graph. If you properly do it, you'll have a stack of them. So here we came from down, and then from here, and 2 and 1. And if I'm done with this, the numbers on the edges will form an Euler cycle. 1, 2, 3, 4, 5, 6, 7, 8. Okay? And it's not too hard to prove that this always works. I'm not going to do it in more detail here. The code is very detailed. The key thing is it's essentially a depth-first search. The difference is we're not stopping visiting a vertex once we've seen it for the first time. It's an edge-based depth-first search. So I will revisit vertices as long as there's edges I haven't seen. So I have a visited array, which here is 2-dimensional. It has an entry for every possible edge. This could be a map from edges to true, false, just to write it more concisely. Otherwise, it's a standard depth-first search. You have a stack of vertices where you came from. In each point, we start with putting some start vertex on the stack. Here we have to make sure we use one of those odd degree vertices if there is one. Otherwise, any vertex can be used as a starting vertex. I'm remembering for every vertex in the graph, what is the next edge to look at when I'm visiting that vertex again. So I'm remembering an iterator in its adjacency list. And initially, that's just pointing to the first edge of every vertex. Then I'm calling the recursive depth first search. It's with an explicit stack, sorry, nothing recursive. It's the iterative version. I have my start vertex that initially goes onto the stack. And then, as long as there are things on the stack, I'm not done yet. I get the topmost vertex and look at the next edge from that vertex that is not yet processed. If there's none left, that's the backtracking case. If there is one left, then I'm following that edge. So there's again the case. Sorry, that's the case. I follow the edge. If it has a next edge, then I'm following that edge. I mark the edge as visited, and because it's an undirected graph, I mark it visited also in the backwards direction. And then I'm pushing the vertex I arrived at. This is the backtracking case. If a vertex I reach doesn't have any more outgoing edges, I have to go back from where I came. So I'm popping the last vertex from the stack, that's where I came from. And I'm writing in this Euler array at the last position, J starts at M. I'm putting in the edge that I've used here, that I've used to get here. Okay, that's the same algorithm I've showed in the example, just explicitly written with a stack as you would for a depth first search. All of this was for undirected graphs. That's how you usually introduce Eulerian cycles. For our De Broglie graph, we need this in a directed graph, which maybe sounds like we have to do all the work again, but there's only very minor changes that are needed. First of all, Euler's theorem, if you think about the proof that I skipped, the directions are both simple, and if you take them separate, is very easy. If we have the second condition, then we've just listed an algorithm that computes an Euler walk, so certainly one exists. So that direction is given by the algorithm. The other direction is easy. If it has an Euler walk, you can just observe that apart from maybe the start and end point, every vertex in the middle is entered and left the same number of times. So it has to have an even degree in this undirected case, right? In the directed case, something similar is still true. If there is an Euler walk that is directed, well, every non-start or end vertex still has to be entered and left the same number of times. So its total degree will have to be even, but actually because the edges have directions, it has to have the same in and out degree. So it's balanced. The connection here is the only reason why this is more complicated is you can't concisely say the number of odd degree vertices is 0 or 2, because for the start and end vertex, you either have one more incoming or one more outgoing edge. You need one of the two types. So you can lift all the theory to directed graphs, and the same kind of proof works. And the same kind of algorithm works to compute the Euler walk if the right-hand side is fulfilled with minor adjustments. Initially, when we started here, we looked at the degrees of vertices and then picked one that has odd degree if there was one. Also, if we're not in the case that the graph is Eulerian, we just stop. We can do the same here using this condition. The start vertex has to be the one that has more outgoing than incoming edges, also obvious. So the only non-trivial change is at this point where we mark edges true, where we mark that an edge is visited. because it's directed, they're not the same, so we don't do this. This is left out in the directed case. All the rest of the algorithm is the same. That's the beauty of going once through the pain of writing down a depth first search version. Then the directed case just pops out as unchanged. Okay. I went through this fairly quickly because I think everyone will have seen that at some point in their life, but it might be a while ago. So also the directed version is often treated a bit stepmotherly. Like for some reason, this is not receiving the same amount of love, probably because of the historic stories. Also, it's fairly easy. Now let's do a little bit of a detour on the analysis of the algorithm. How efficient is the algorithm? It's a depth-first search, even though it's edge-centric. It essentially visits everything in the graph once, but it visits every edge at least, at most once. But the running time otherwise is actually the same as a standard depth-first search, because that also visits every edge. It just doesn't traverse it and moves on. It just looks over the edge and says, the target I've seen before, I'm not going to go there again. But you're still going to have running time proportional to the number of edges as well. The same is true here. You'll essentially treat every edge once. Fair enough, maybe twice, because you also mark it with an index. But a constant factor aside, the running time is the same. Now, what's more expensive in this implementation is the space that we use. At least in a graph where there's many more edges than vertices, the data structure for the stack here, this frontier of vertices that we're still actively exploring, this may contain the same vertex many times. I don't know if this is obvious from the quick pace I went over this. It is certainly obvious from the example we've done that we revisit vertices. So the condition here is I may have a vertex any number of times on the stack, as long as the stack forms an edge simple walk. So I'm not going to have a vertex more copies of a vertex on the stack than it has edges. So in total, the size of the stack is bounded by the number of edges, but it may be much more than the number of vertices if there's more edges than vertices. Okay? So worst case, you're walking a very long path and essentially using all the edges, then all of the copies of the vertices are on the stack simultaneously. And so as a fun little detour of some work we've done in my group, we asked ourselves, is this needed? We're doing a depth-first search, even though a modified one. Do we really need M words of space? Or can we get rid of this dependence on the number of edges and go back to what standard traversals have, M plus N time, but N space, because every vertex is at most once on the stack in the standard DFS. And the answer is yes. That's a paper coming up in January on the Symposium on Simplicity in Algorithms. So it's not just supposed to be a new result, but also a very simple algorithm. So I thought we might discuss it here briefly. And the key observation is that really, for Euler walks, you don't actually need the full power of a depth-first search. If you think about it this way, again, my tiny example from before. There's another vertex. I can basically walk whatever. When I'm stuck, I'm just going to revisit some place where I can branch off and insert that into the current tour. So I could have said, what do we do? This one. Here I was stuck. The red tour is done. But I could have branched off here with the blue tour, which is a cycle. And it necessarily, let me try it a little less confusing. It has to be a cycle because after I've taken out, the red one is a path. So it's taken out all the odd degree vertices. What's left is only even degrees. So any Euler paths, Euler walks in the remaining graph will have to be cycles because there's only even degrees. And so I can take the red tour up until this point, then take the blue detour and then continue. And that always works. Now, if I have several of these blue cycles, any combination, any ordering of inserting these other cycles into the red path will be a valid Euler walk. It doesn't matter. I don't have to remember in what order I found them. Now, a depth-first search very carefully keeps track of the precise order in which I found all those edges, and the Euler walk just doesn't care. So let's not do that. Let's not store the exact order in which we found edges. But you still have to make sure you don't get stuck in the final backtrack. So you need to store the first edge you reach a vertex with separately, but then all the others, you can just say, yeah, I've seen those. The order doesn't matter. Let me show on a short example how this works. And I couldn't convince my co-authors to convert this to something that I found more intuitive. So we'll do the whole algorithm backwards, using all the edges backwards, with the benefit that you can produce the Euler walk in the right order. Whereas here, remember, here Holzer's algorithm here, a while ago. Where's the algorithm? It essentially produces the Euler walk backwards. It produces the last edge first. And here I reversed it by putting the first edge I find at the end of an array. If you don't want to do that, if you want to output the walk in the correct order, you can just work on the reverse graph, where you're using all the edges backwards, and then the reversal is not needed. I find this confusing, but it also works, of course. Initially, all the edges are black. And I'm going to encode what the algorithm does with two more colors. The red edge of each vertex is the first time it's reached in the traversal. And then it might be reached many more times with other edges. And these I'm going to also mark as visited, but in green. And then the order among the green edges is irrelevant. And the second important thing, whenever I backtrack, I have to prefer green edges over the red one. Let's see in the example, and I think you'll see how this maps to the original algorithm. We're starting at this vertex. Now remember, I'm doing everything backwards, so I have to go to 6. And this becomes a red edge because it's the first edge that reached 6 ever in its life. Then I'm continuing from 6 to 5. This becomes another red edge for 5. Now here I have a choice, right? There's 2 incoming edges, but I'm going to 2. And then from the 2, again, I could have gone back, or I could go to the 1. Let's go to the 1. Vertex 1 I've seen before as the start vertex. So this edge is not red. The red edges always form a tree, the DFS tree, in the standard sense. But now I have this other edge. I mark it as green, as visited. Now at vertex 1, it's the first time I have to backtrack. There's no more edges I can explore. Now this is where this green preference rule comes in. I have to follow the green edges. So backtracking usually takes the edges backwards, but here, because we're working reverse, we're actually taking the green as the first. And so this will become the first edge in the final Euler walk. Okay? And so we mark it as such. It gets the label 1 just for making the example easier to follow. And we mark it as dashed because this edge is essentially gone. We don't look at it ever again. Then we continue. We're back to vertex 2. Are there more edges I could have followed? Incoming edges, yes. I can go back to vertex 5. This is a green edge because I've been at 5 before. Then here I can continue up and up and up. So here, another incoming edge, but I've seen 2 before, so that's going to get a green again. Now I've seen all the edges, so the rest will be all backtracking. And all I have to make sure is that I prefer green edges if there are any. Here, there is a green edge to backtrack over. Here, there's no choice, so I just do whatever. Follow the edge. Here, I'm going up for the green first, and then red, red, red. Notice that there will always just be one outgoing red edge by construction, because it's the edge from the DFS parent. There's only one ever. But green edges, I may have many more. In the tiny example, I never really had 2 green edges, but to make it fit on the slide. And then I have my Eulerian walk here in this graph. Okay. Now, why did this reduce the space? This is from the colors, at least you can say I'm using m bits or order m bits to store the colors. But you actually don't need that. You can store everything implicitly under the assumption that the input graph is represented in a way where I can iterate over incoming and outgoing edges. You can debate this, but many representations of graphs do this, and many libraries implementing graphs actually explicitly duplicate directed edges because they want to support this traversal in both directions anyways. So I'm just assuming that this doesn't cost me extra space. But yeah, I can justify that. And as I said, every vertex has at most one outgoing red edge by construction. It's a DFS tree. So this one I'm going to store explicitly. I'm going to keep for every vertex an entry that says which was the red edge that I've reached it via. And then the other edges that are visited, which means they're red or green, I don't even have to store this at all, because I'm, like in the previous code, I'm just storing a pointer, an iterator in the adjacency list of a vertex. And so all the edges that are after the iterator are not visited yet. All the edges before have been visited. And I never really have to ask, is an edge visited or not? I just follow the iterator. So this costs essentially nothing extra in space. So all I need to store is the red edges and the iterator for the vertex, and whether I've visited a vertex before. So this is all things that just take linear space in the number of vertices. Maybe this might be good to say at the end. Okay. Give me 1 minute to wrap this up. If we look at where we came from in genomes, there's a few things that we haven't discussed in detail. The Eulerian walk might not exist because for some reason I have errors in my input. Or if it's not unique, we may disqualify the graph as having a single walk. More likely is that my input is just not a perfect k-mer composition. And then I still want to do something, and luckily I can still construct this De Bruijn graph and at least find what bioinformaticians then call contigs. So it's sequences of contiguous ranges of the genome that you can sequence. Even though you don't get the full genome in one go, you can get larger fragments, which are much larger than the initial reads. And it seems that this is something people can then manually work with much better. And we can get this from essentially looking at in and out degrees. This is just a summary of some challenges in practice. These are essentially for your interests. So we don't really have a single string. It's broken up into chromosomes. So we certainly want to do one chromosome at a time at most. Most of us have two. So us in some sense of animals or eukaryotes maybe more generally. We usually get chromosomes from the two parents. And they're very similar. So this is actually horrible for sequencing. So the human reference genome is actually just one of the two chromosome pairs. And this seems to be acceptable. We might have gaps in the coverage of reads. That's a bigger problem. And we may have errors in the sequencing. And then another beautiful thing is the double-strandedness. So we get essentially not just one text, but the text and its reverse complement. and we'll get k-mers from both strands mixed. So that's another little puzzle to handle that part. There's a few things you can do. Imperfect coverage is relatively easy to address. If there's some starting positions you don't have, but you have long enough reads, you break them into shorter k-mers, and then artificially you get much more reads than you started with. So this, I thought, was really hard to imagine, that the molecule is really broken up at every single possible base. But that's not at all an issue. You could just simulate that by making the reads a bit shorter and using all the possible starting positions within the reads. And the chromosome pairs also seem harmless. You can, in the lab, make sure you only sequence one. The more problematic thing seems to be the read errors. And the De Broglie graph completely dismisses, if the overlap is not perfect, it completely says no edge, go away. And that seems to be the major trouble, which is why people in practice tend to use the overlap graph. Because there, it's much easier to incorporate inexact overlaps, give them a weight, and then compute essentially a traveling salesperson tour of minimal cost. Whereas the De Broglie graphs with the lovely Euler walk theory seem less amenable to this generalization. Okay, good. That's all I wanted to say about the genome sequencing. I think it's a cute example of how the first obvious formalization of the problem runs into a hard problem, and then if you look at it differently again, it's actually a tractable problem. Maybe the lesson there should be, just because something looks NP hard in the first attempt doesn't necessarily rule out an efficient algorithm if you frame the problem differently. All right. See you for the tutorials tomorrow. And we'll try 2 p.m. next week. And yeah, you'll check out the recordings. Okay, let's do it a try, and I'll keep the block on this room so we can come back to the original slot as well. Oops.